Okay, can you see my mouse?
Yes. Yeah.
Oh, cool.
Great. Okay.
Cool. So, hi everybody. I'm very happy to present you our work today. We are working on the mutations of a sodium channel. Some of those mutations, they cause migraine. Some of those mutations, they cause epilepsy. And we are studying them with a modeling approach. But we are working with Massimo Mantegazza, who is a neurophysiologist in the IPMC lab. So with his team, they are doing experiments on mice. First, we will see what is known about the mutations of this channel and what remains to be understood. Then I will present the mathematical model that we developed to study that. And then what we can learn from this model concerning the migraine mutation and concerning the epilepsy mutation. So NAV 1.1, it's a voltage-gated channel for sodium ions. It's mainly expressed in GABAergic neurons, which are inhibitory neurons, and it's very important for the excitability of those neurons. Several mutations have been identified for this channel, and some of them, they cause a disease called familial hemiplegic migraine type 3. That's a subtype of migraine with aura. With aura means that you have sensory disturbances before or during the headache. And the common point of all those migraine mutations is that they cause a gain of function of the channel. So the activity of the channel is enhanced. On the other hand, other mutations of the same channel, they cause severe childhood epilepsy, for instance, Dravet syndrome. And contrary to the migraine mutation, those are loss of function mutations. And in both cases, the link between the mutation and the disease is not completely understood. In the case of migraine, we know the end of the story. We know that the headache is caused by something called cortical spreading depression. That's a wave which propagates in the cortex. Here you can see the recording of one neuron. So first there is a high firing frequency of the neuron, and then a plateau, like at voltage which is larger than the resting potential. And this plateau here is called depolarization block. All the neurons, they are doing this one after the other, and on the right in white here, you can see the propagation in the cortex. Those are pictures of a brain slice of a mouse. Those data, they are from the neurophysiologists we are working with. And so when the wave reaches the meninges, it will indirectly stimulate some pain receptors, and this is what will cause the headache. So we know the link between the CSD and the migraine, but we don't know the link between the mutation and the CSD. And what will interest us is the initiation of the CSD rather than the propagation. Now I will present you the mathematical model that we developed to study those mutations. It's a conductance-based model, so like Hodgkin-Huxley is also a conductance-based model. We consider a pair of neurons, a pyramidal neuron and a GABAergic neuron. And so the pyramidal neuron is excitatory and the GABAergic neuron is inhibitory, and we implement the mutation in the GABAergic neuron. And in our model, we take into account all the transporters which you can see in this diagram. In Hodgkin-Huxley, we have some which are like in Hodgkin-Huxley, for example, the voltage-gated channel or the leak currents. But we have other transporters which we need, for example, the sodium-potassium pump, which is needed to restore the ion gradients. This model is based on a model which has been published, but we make several changes to adapt it to our situation. So the first change, of course, is to implement the migraine and the epilepsy mutations of NAV 1.1. And I think it has not been done before to model both of those mutations within the same model. Something important is that in our model, we have dynamic ion concentrations. Often the ion concentrations and so the reversal potentials, they are assumed to be constants for simplicity, but in the case of migraine or epilepsy, we cannot make this assumption because the ion gradients, they are completely disrupted during the attacks. So our neurons, they are going to be coupled not only through synaptic connections, but also through the variations of extracellular ion concentrations. And the last change which we made is to replace the model used to describe the gating dynamics of the voltage-gated channel of the GABAergic neuron with the more recent model, first of all, and which is specific to the cortex. Why do we want it specific to the cortex? Because there are some experiments in which a toxin is used to mimic the effects of the migraine mutation. And with this toxin, we can see spontaneous CSD, but only in the neocortex and not in other brain areas. Now I present you how we model the mutations. So NAV 1.1, it's a voltage-gated sodium channel. At rest, it is closed, and when the voltage increases, it will open. But then there is an inactivation process which makes it close even though the voltage is still high. And those activation and inactivation process, they create transient sodium currents. However, if there is something wrong with the inactivation, then there is persistent sodium currents. You can see here the equations for this. This variable H models the inactivation, and in the case of persistent current, it's equal to 1. We don't have any inactivation. And persistent sodium currents, it's the common point of most migraine mutations of NAV 1.1. So to model those mutations, we partially replace transient current, yes, with persistent current. To model the epilepsy mutations, those are loss of function mutations, so we simply reduce the maximal conductance of the transient current. In our model, there are some conserved quantities, and this is a consequence of the fact that we have dynamic ion concentrations. Because as you can see here, for each current, it will appear both in the equation for the intracellular ion concentration and in the equation for the membrane potential. So from because of that, this expression here is the first integral in our system. It's constant with time. Okay, it's not a problem, but we have to be careful with the way we model external inputs to the neurons. If we would use a constant current which appears in the equation for the voltage, if we would do this, then instead of having a constant here, we would have constant plus something which grows with time. So we have a drift of the system. We cannot have steady states. We cannot have limit cycles. So we don't model external inputs like this. Instead, we use excitatory synaptic currents. When we want to stimulate the neuron, we say that there is a constant glutamate input on AMPA receptors, which are those receptors here and here. And this recreates sodium and potassium currents, which can appear in the equation for the ion concentrations. So we don't have a problem of drift of the system. And the parameter which we modify when we want to stimulate more or less the neurons, it's the conductance here. I will not enter into the details, but those are the equations of our system. There is 14 variables. It's a lot, so it's not so easy to study this model. We could remove 2 equations because of using the first integrals, which I mentioned earlier, one for each neuron. Okay, so that is our model. Now I will present you first the results concerning the migraine mutation and then the results concerning the epilepsy mutation. First, we focus on the effect of the mutation on the GABAergic neuron itself. So for now, we removed the interaction with the pyramidal neuron. And we want to see the effect of the mutation on the firing properties of the neuron, in particular firing frequency. So we start from neuron at rest, and then we give it some external input, and we count the amount of action potentials which are generated in a given duration. But we don't really see an effect of the persistent current. However, what we can see is that when we have more persistent currents, then the firing of the GABAergic neuron releases more potassium in the extracellular space. So this was quite surprising when we talked with the neurophysiologist, Massimo Mantegazza, because he was surprised that we can have more release of potassium, but not really an effect on the frequency. This is possible because simply the amount of potassium released at each action potential is larger. What is going to be the effect on the initiation of CSD when the two neurons are coupled? We are going to say, we define that a CSD is initiated when there is a depolarization block in the pyramidal neuron. Migraine, it's an episodic disorder. This means that you have symptoms only during the attacks, and otherwise everything looks normal. So there is a trigger factor which will cause the transition from the physiological to the pathological state. And we will see that persistent current, it reduces the threshold for this transition. Let's look for now at the curves in green. It's with the default parameters. Here on the left, you can see the minimal external inputs, which is necessary to initiate a CSD. And you can see that this decreases when there is more persistent current. Moreover, when for a given large external input, the CSD is initiated earlier when there is more persistent current. We also tried to reduce the diffusion of extracellular potassium. This can model in a very simplistic way the contribution of other GABAergic neurons, because in our model we just have one GABAergic neuron. Or it can model a failure or less efficient glial network, because the glial cells which are around neurons, they have this role of buffering the extracellular concentration. And as expected, we can see that when we have less diffusion of extracellular potassium, the threshold for CSD is reduced, and CSD happens earlier for a given drive. This is consistent with the experimental data. So they are using a toxin to mimic the effect of the mutation on the channel. And then they see that when brain slices have been bathed with the toxin, they can see spontaneous CSD, which they never see in the control conditions. And this happens only in the neocortex. They also use optogenetic to stimulate the GABAergic neurons, which corresponds in our model to a strong external input. And they see that when they have the toxin, then the latency for the CSD is reduced. So it's consistent with what we have. Now let's see some traces of our model. So on top, it's without persistent current, so the wild type case. The two neurons are spiking and the extracellular potassium remains at a reasonable level. Here you can notice that there is a short delay before the pyramidal neuron starts to spike. This is because in our model, there is an inhibitory synapse from the GABAergic neuron to the pyramidal neuron. When we have the mutation, the traces, they look quite different. The two neurons, they enter depolarization block here and here, and there is a huge increase of extracellular potassium. Let's focus on the beginning of this simulation, the 10 first seconds. You can see the instantaneous firing frequency of the two neurons and extracellular potassium. And you can see that with the mutation, the potassium increases faster than without the mutation. This makes sense because we saw that the effect of the mutation is to make that the firing of the GABAergic neuron releases more potassium. Then at some point, the firing frequency of the two neurons increase a lot, especially for the pyramidal neuron. And then here they enter a depolarizing block, so they don't produce action potentials anymore, while the potassium continues to increase. So in our model, the mutation facilitates the initiation of CSD, and the important thing seems to be the accumulation of extracellular potassium. Now we move to epilepsy. So like for the migraine, we first studied the effect of the mutation on the GABAergic neuron itself. We start from neuron at rest, and we give it some external inputs, and we count the amounts of action potentials which are generated. And we can see that when there is the epilepsy mutation, so reduced sodium currents, the number of action potential reaches a peak and then starts to decline. We understand what happens if we look at the traces. In fact, when we have the mutation, the GABAergic neuron here is entering depolarization block, so it does not produce action potentials anymore. What is going to be the consequence on the pyramidal neuron? Well, when the GABAergic neuron enters depolarization block, it suddenly stops to inhibit the pyramidal neuron. Here, this is what is happening here. The GABAergic neuron stops to spike, and at the same time, it's not properly speaking a seizure, but there is a sudden increase of the firing frequency of the pyramidal neuron. Okay, so to conclude, we could capture within the same model migraine-like and epilepsy-like behaviors. Concerning migraine, the main effect of the mutation is to increase the release of the potassium by the GABAergic neuron, even though the firing frequency is similar. And this accumulation of potassium outside of the cell, it will increase the reversal potential for potassium for the pyramidal neuron, which facilitates the initiation of CSD, which is going to cause the headache. For epilepsy, the effect of the mutation is to make the GABAergic neurons, according to our model, of course, more susceptible to depolarization block. So when this happens, the inhibitory restraint they have on pyramidal neurons is suddenly removed, and this is favorable for seizures to start. Of course, this should be tested experimentally. So in particular, what is interesting is the fact that we have increased release of potassium, even though the firing frequency is similar. And I hope the experimentalists we are working with can find some experiments to test this. What we are going to do next is to reduce the model, because as you could see, this is a huge model. We have many variables, many parameters, and several time scales, so it's not so easy to study this model from a mathematical point of view. We would like to get a simpler version of the model while keeping the important features of this one. And with this simpler model, we would like to study the bifurcation scenarios for the transition to migraine and transition to epilepsy. Okay, that's it. Thank you.
Thank you very much, Louisiane. Very, very interesting talk, very nice study. And I'm curious to see how the experiments will turn out and whether your hypotheses can be answered. Yeah, there's plenty of time for questions. And it's again Emre who starts off with the first one. Thanks, Louisiane, for your nice talk. It was very informative. I would like to ask the following. For both migraine and epilepsy, the model uses the same variables, or some of them are ignored or removed from the model depending on migraine or epilepsy?
No, we use the same variables in both cases. It's just that for migraine, we change. In fact, what we change is only the parameters for the maximal conductance of the transient current and persistent currents. For migraine, we partially, we keep the sum of those conductances constant, but we add some persistent current and we remove transient conductance. And for epilepsy, we just reduce the conductance of the transient current, but all the rest is the same. I don't know if that answers the question.
For me, it was very clear. There's a thumb up. Yeah. In the chat, Emre says it's definitely was clear. Hilmaya asks another question. Interesting. In your results, you present mainly potassium. What about chloride and sodium? Do they show any interesting behavior?
Okay. Yes, they also vary a lot. during the simulations, but not as strikingly as the potassium. But they are also doing, they are changing a lot, and for sure it would be interesting to study that, but we focused on the potassium.
Okay, if there's more questions. Yeah, Maya quickly responds to your answer and asks whether you could also set them to a constant.
Let me think. I don't think so, because, well, I don't know. For example, the activity of the, we have this sodium-potassium pump, which restores the iron gradient, and its activity, it depends in our model on the intracellular sodium concentration and potassium concentration. Okay, we could have constant sodium. Okay, I don't know, in fact.
Yeah, I believe that will be one of your major challenges when you try to reduce this very detailed model too late, to make it mathematically more amenable.
Yeah, when we reduce it, in fact, we don't want all those equations. We are going to have to make some choices and maybe keep the potassium and not the other ones. So I guess, yes, it should be feasible.
Yeah, thanks a lot.